You may or may not know about the work that we've been doing with a compound called CuATSM. This is the structure of the compound on the screen. Highlighted in the middle is a copper atom. You'll hear me say the word copper during this talk many times. We've been putting the compound into SOD model mice for quite a few years. And this is just an overview of some of the outcomes that we generate. The slide on the left, I just want to highlight that even though the initial work that we started doing, we were doing relatively simple treatment paradigms, pre-symptomatic, long duration treatments, we've ultimately come to a treatment paradigm which is relatively aggressive paradigm in that we are treating pretty late in symptom development in these animals. So just to highlight where we are starting our treatments at the moment, it's well after a peak body weight, and it's at about the stage where you're starting to see a decline in the rotarod test for motor functionality. So in this example on the left, we're treating about a week before you see an overt decrease in the motor phenotype, but in some of the studies it's at about exactly the time when you do see that overt decrease on the motor test. I'm sure if you did a much finer test for motor function, for those of you who know, the rotarod test is a pretty crude assay. If you did a finer test, I'm sure we're treating well after the onset of some finer motor symptoms. And the graphs in the middle and on the right are the sorts of outcomes that we've been generating. A survival curve needs no explaining. The outcomes are pretty robust. And in terms of motor function improvement, as you can see in this graph on the far right, we're treating pretty much as the symptoms start on the motor function test. So there's no effect in shifting onset, obviously, but the rate of decline in motor function is substantially delayed. These are some of the publications that we've generated along the way. And the drug is orally bioavailable, so all of this administration to the animals is by gavage. Now this is another set of data just showing dose response of the compound. These treatments were done in the, for those of you who know, the price for life paradigm, in that it was the SOD G93A animals on a mixed genetic background, and treatments for this study started at 50 days. The thing that's particularly exciting about this result is reproducibility across multiple institutes, multiple countries, different investigators. We saw the long list of failed compounds before. One thing that is persistent issue in the field is lack of reproducibility of outcomes in animal models. So while many drugs will fail in the clinic, you can make the argument that many of them were never going to be reproduced anyway. So to get this sort of independent validation, I think is pretty good endorsement for CuATSM in these mouse models. So that begs the question, obviously outcomes in a mutant SOD mice may or may not have relevance to people in the clinic, may or may not have relevance to people who do not have a mutant SOD form of the disease. So whether or not the compound is going to be relevant to people who have ALS, specifically people who do not have a mutant SOD form of ALS, that's the major outstanding question that we're trying to answer at the moment. So I'd just like to point out that the therapeutic outcomes for CuATSM in mice are not restricted to mutant SOD1 mice. In parallel, in Melbourne, we've been doing work in Parkinson's disease models. We've done some work with collaborators in Finland working on a stroke model. And in all of these disease models, CuATSM is protective. We've also got some additional unpublished data from other models of neurodegeneration in which CuATSM is protective. So at least in the animal models, the neuroprotective activity for CuATSM is absolutely not restricted to mutant SOD1. And also, even if you do look within the mutant SOD mice, the sorts of readouts that we're getting for neuroprotection are pretty broad in nature. Protection against oxidative and nitrosative stress, protection against a form of cell death known as ferroptosis, and also protection against toxic astrocytes. This is something that's becoming absolutely much more compelling in terms of a drug target. It's been known for quite some time that astrocytes do play a role in neuronal death. Can we target them specifically? There's evidence now that CuATSM absolutely is mitigating neurotoxic astrocyte activation. Now, I do not want to steal Jeff Liddell's thunder, but I do want to put in a quick plug for his poster. He'll be talking to this poster tomorrow. Please come along and have a chat. And also work that's completely independent to ours is the work that's been done by Cassandra Denise Rivers, where she has also been putting CuATSM into astrocytes derived from patients and showing protective outcomes there. But the mechanism that I'm going to talk about today is the one that involves improving copper bioavailability. We did a study a few years ago where we showed that when you administer CuATSM orally to mutant SOD1 mice, if you were able to track the copper from the compound, you could eventually detect it on copper requiring proteins in the central nervous system. So the compound is getting into the central nervous system, it is making the copper that it delivers bioavailable, and it's entering the natural requirement for copper in the central nervous system. This graph here, it doesn't matter if you don't understand what it means. Basically, the red trace, which is dipping down, is showing copper being transferred from the compound to copper-requiring proteins in the central nervous system. So if I'm going to talk about a mechanism of action that involves improving copper bioavailability, the obvious question is there actually a copper problem in human ALS, particularly in sporadic cases of human ALS? Now, you might not actually be interested in whether or not there's a copper problem in human ALS. I just want to point out that copper is an essential micronutrient. Basically, if we don't have enough copper in cells in our body, we just don't live. It is an essential micronutrient because it imparts activity to some very important copper-dependent enzymes, not least of which is cytochrome C oxidase of the mitochondrial respiratory chain. If you don't have copper on cytochrome C oxidase in the respiratory chain, you don't generate enough ATP to live. So that's the significance of cuproenzymes in the eukaryotic system. And of course, there's a raft of other copper-dependent enzymes. Now, if you just take a human spinal cord, homogenize it, and measure the amount of copper in that spinal cord, you're not going to see much happening. This sort of data has been published before, and these are our data from human spinal cord tissue. No immediately obvious change in copper in this tissue. But if you get a little bit more sophisticated with your analysis, you do start seeing things happen. So this technology here, it might look like a standard microscopy stain that you're looking at, but these images for copper in the human spinal cord, the way we generate these images is that we take the tissue section on a standard microscope slide, we put it under a laser which ablates the material from the slide. The material then goes into a mass spectrometer. The only difference between the mass spectrometer that we use and the standard mass spectrometer is that ours measures elements. So of course we measure copper. And by using the laser to ablate the material into the mass spec, you can retain that spatial information and you can reconstruct the 2D image. So these 2D heat maps that you're seeing represent the abundance of copper in the human spinal cord. And you can see the difference quite clearly between the control cord and the ALS-affected cord. Now, this is a sporadic ALS-affected cord, by the way. All of the human tissue analyses that I'm going to show you come from sporadic cases of the disease. And of course, once you've captured this information, you can do a region of interest analysis, and you can see if you just measure copper in the total grey matter area, you can see a decrease, and you can see a marginal increase in the surrounding white matter. So what we're seeing here indicates that there's not an overt copper deficiency in ALS, but what you do have is a perturbation to the natural distribution of the copper. And if you take those same spinal cord samples, homogenize them, and do a relatively crude biochemical separation into soluble and insoluble, it really doesn't matter what soluble, what it's soluble in. In this instance, it's TBS. But if you're just partitioning into different biochemical fractions, it could be a membrane fraction, a nuclear fraction, you can then start looking at the amount of copper in these individual fractions. And you can see here there's a decrease in the soluble fraction and an increase in the insoluble. So again, indicating that it's not an overt copper deficiency problem, but there's an imbalance and there's a redistribution problem. This image on the left is indicating that there's a bunch of different copper chaperoning transporters, chaperones, because getting copper into a cell and then making sure that it goes to where it's needed, it involves a pretty complex cellular machinery. We've developed a system, a qPCR system, where we've developed our own cassette for looking at these particular copper-related genes. And you can see in the human ALS-affected spinal cord, a fair chunk of these genes are actually shifted in the ALS. And if you do the same analysis of the mutant SOD1 mice, the spinal cord there, you can see that a lot of these genes are also shifted there. So there's a lot of gene expression changes related to copper in the human cord, and many of those are actually recapitulated in the mutant SOD1 mice. And in terms of functional consequences, we're still working through the list of enzymes that require copper, but what we're doing is we're discriminating between the abundance of these proteins and their copper-dependent activity. Here is a decrease in peroxidase activity. I'm going to come back to this one at the moment. In the central nervous system, most of the peroxidase activity comes from a glial protein known as ceruloplasmin. You can see it's a fairly substantial decrease in peroxidase activity in ALS. Dopamine beta hydroxylase, you do an initial analysis and there's no clear difference. We measured the protein amount for DBH and there's actually a slight increase, but the activity is not going up proportionately, suggesting that there's an accumulation of copper-deficient DBH. And the DBH assay is actually amenable to supplementing with exogenous copper. And if you do that, you can see that you do get an increase in activity when you supplement copper into the control cords. You get a higher response when you supplement it into the ALS cord, providing further support for the idea that you do have an accumulation of copper-deficient DBH in ALS. And I've mentioned a couple of times now that it's not an overt deficiency problem. It's a partitioning problem. It's the fact that the copper is there. You measure total copper in the cord, remember, and there's no change. The copper is there, but it's just being shifted around and it's getting to different places. So here's an example of lysyl oxidase activity is actually increased in the sporadic ALS spinal cord. So maybe it is that the copper that's going to the LOX is coming at the expense of copper on peroxidase, on the ceruloplasmin peroxidase. So the peroxidase is particularly interesting to us at the moment because we're now seeing all of these copper-related changes, but the question is, so how does this contribute potentially to neuronal death in the disease? And this is one of the pathways that we're particularly interested in pursuing at the moment. Basically, peroxidase functionality within the central nervous system, within the glial cells, the peroxidase ceruloplasmin, what it does is it, when it's at the plasma membrane, it stabilizes ferroportin at the plasma membrane, and it also imparts the redox activity that's required to actually export iron from the cells. So if copper-dependent peroxidase activity is decreased in ALS, you can expect that there's going to be an effect on iron, and this is what we see. In the same spinal cords where we're seeing a decrease in copper-dependent peroxidase activity, we're seeing an accumulation of iron. And when you manipulate copper availability to glial cells in culture, you actually see a pretty strong correlation between cellular copper levels and cellular iron levels. Now, I mentioned Jeff Liddell's poster before. If you want the detail, please go and speak to Jeff. I'm going to summarise a few years' worth of work in this one schematic. Basically, what we're seeing now is we're making this connection between altered copper availability in ALS to a form of stress known as ferroptosis. The ferroptotic stress is an iron-dependent form of stress. So we've got this connection between the copper availability and the ferroptotic stress. What we've got, and what Jeff will show you about if you go and have a chat with him, is the cascade of events that ferroptotic stress triggers in ALS. Basically, we're seeing this cascade of events initiated by a ferroptotic stress affecting microglia, which is then activating the astrocytes. The astrocytes are becoming neurotoxic, and the neurotoxic astrocytes are killing the motor neurons. And of course, if you come in with CuATSM, you can inhibit this entire pathway. So this might not be the definitive answer for how CuATSM might be a therapeutic option for ALS, but it is absolutely a pathway which, you know, for sure, if this is all happening in the human cord, this would be beneficial to hit with a compound like CuATSM. So in summary, yeah, for sure, CuATSM is very protective in the mutant SOD1 mice. We're developing a data set that suggests that the mechanism of action, at least in part, involves augmentation of copper bioavailability in the spinal cord and the brain. What I've shown, very briefly at least, is that when it comes to the copper biology of ALS, there are plenty of indications now that copper is disrupted in ALS, in sporadic ALS. Importantly, many of these features, we hear much talk about how much the mutant SOD1 mice may or may not be a pertinent model for drug development for human ALS, especially sporadic ALS. But what we're seeing is that on the copper front, many of the features that are evident in the human affected cord are actually recapitulated in the mutant SOD1 mice. So just to wrap up, CuATSM for sure is not a conventional drug. It's not the norm to put a copper-containing compound into the human central nervous system. Copper is also not a conventional target. But what we've developed is a body of data that says, yes, copper perturbations are absolutely present in sporadic ALS, and with the CuATSM at least, it's a feasible therapeutic option. Phase 1 testing was completed relatively recently, and a recommended phase 2 dose was identified. It was a very successful phase 1 trial. There were some positive indications for therapeutic efficacy. Hopefully those positive indications will be borne out in the phase 2 trial, which has just started recruiting patients. And that's me. I think it's time for beers. I will mention very briefly James Hilton, Jeff Liddell and CuATSM. So while this project has involved a lot of people and it has been fortunate enough to get good support from funding agencies, the people who really have generated all of the outcomes are Jeff, James and Kai. Thanks for your attention.